So today we are looking at the fluid mosaic model. What is this sort of concept of fluid and mosaic, and how does this help us understand the cell membrane? So if we think of the name of this model, we have the fluid mosaic model. Okay, so a model is very simply a way to represent something. So it's a simplification to represent. And in this case, we're looking at the cell membrane. So why is it fluid and why is it mosaic? And it's important to get this stuck in your head because they like to ask these questions. So fluid makes us think, I hope, of liquids, of movement, and that's why we're using it, okay? The membrane is fluid, it's dynamic, it's changing, things are going on. And so both the phospholipids and the proteins can move around. Okay, so that's within the bilayer. So the proteins that are on the outside layer will move around in there. The phospholipids will move around in their layer. And then on the flip side, you've got the proteins and the phospholipids, and proteins that go across the entire thing, they can also move. So although we look at set diagrams and set structures, nothing here is set. Okay? It's an actively moving situation. And when you think about it in terms of bulk transport, your exocytosis and your endocytosis, and it starts to make sense, you know, we've got membranes fusing, we've got membranes pinching off. All of these things must be able to move and adjust depending on what's happening. So don't think of your cell membrane as something static. Think of it as something that's moving and where the components can move around depending on what's happening and what they need to do. Okay, so fluid, we can move around, and mosaic, we have scattered proteins, different types of proteins. So different types means different structures, means different functions, okay? And that is then throughout the membrane. So now you've got to think of the membrane not as something that you cut through, but as a surface. So all through your surface, you're going to have different proteins popping out with their hydrophilic bits on the exterior, for example. Okay, and that different scattering of proteins is what makes it appear sort of like a mosaic. So there's no extra words here. It is a mobile fluid system. We have scattered proteins throughout the membrane, and we're representing the cell membrane with this model so that we can keep track. Right, and we now have everybody here, despite trials of internet and power, and hopefully we'll all be able to stay online for the rest of the lesson. Fingers crossed. Right, so now let's get to actually details of this fluid mosaic model. Okay, we are going to be drawing the whole thing. Okay, you do need to be able to draw the whole thing. You need to be able to recognize it. You need to know what the bits are. Okay, so what we're going to do is work on an annotated diagram. So there's another term for you. So if we start, let's get us phospholipids to start with. Okay, and we talked about these last week when we talked about lipids, about how this phospholipid has hydrophobic and hydrophilic areas, and that's why it's so important we draw it. Circle and two squiggles. Circle representing our hydrophilic phosphate head, and the two squiggles representing our two fatty acid tails, which are hydrophobic. Right, so we have some phospholipids, and then let's say we have a protein here. Okay, so that protein is just in one half of the bilayer. He's just on one side. So he would just be moving fluidly in this layer. Okay, let's say next to him we have a different type of protein. We're going to draw him like this. Now remember, we're drawing this in 2D. We're not going to draw it in 3D. So the thing is that you got to imagine this as rather like a toilet roll. Okay, so that we've cut in half. So it makes like that shape, like a donut. Okay, but it's a tube. Right, so then the bottom would look like that in my rather dodgy representing of this membrane. Okay, so we carry on with a few phospholipids. Then let's put another protein. Because the more proteins we draw, the less phospholipids we have to get through. But we need them there because our membrane is predominantly phospholipids. And that's how we're getting a lot of the control. Okay, that core, okay, the core of the membrane is this piece. Okay, and that is your hydrophobic tails, which means that your membrane has this hydrophobic core, and that's going to present a problem for things that are coming through that don't like hydrophobic. So in other words, hydrophilic things. Right, so here's another protein. He takes up a nice lot of space, and he's representing a different kind of protein. We are going to go into all these types of proteins in a moment. Okay, have I forgotten anything? Oh, we've got to know one more. So let's keep going a little bit further. With our beautiful phospholipids. Okay, and then we're going to put protein here. Okay, so we have our, let's get some labeling going. Here we have our phospholipid bilayer. Whee! Okay, so I promise that says phospholipid bilayer. Okay, and that is then our overall structure. Remember, we're working a surface here. We're just slicing through that surface to look at what's going on. Now we've got various different types of proteins. So this one here is an extrinsic protein. Extrinsic meaning outside. Okay, this one here would be an intrinsic protein inside the bilayer. Okay, this one here is a transmembrane protein because he goes all the way through. Right, so that's our basic structure. Now let's put some more things in. This one here is a channel protein. And this one here is a carrier protein. So these are involved in different methods of transport. So we said at the beginning, we want to look at doing an annotated diagram. And the thing with that is that we're going to add not just labels, but a little bit of explanation, a little bit of expansion. So when you do your study summaries and you do them by diagrams, you're actually doing annotated diagrams. Okay, annotations is not just some fancy words, it's something we do all the time. So carrier proteins, these are the ones that change shape. Okay, they're being used in facilitated diffusion, and also in active transport. So if you haven't got to the methods of transport across cell membranes yet, don't freak out. Okay, bear in mind there are more methods than you learned at IGCSE. Okay, and that's what we're referring to here. Facilitated diffusion is just a special type of diffusion. Okay, so facilitated diffusion can happen through a carrier protein. It can also happen through a channel protein. Okay, because what we've got here is a channel in the middle. It's protected from this hydrophobic core of the membrane. So if we have protection from that hydrophobic core, we can have a hydrophilic channel. So our ions and other small hydrophilic molecules can move through. This aquaporins is quite a nice example of channel proteins because they are special channel proteins just for water. So they'll just fit. They can be very specific, these proteins. Remember, structure leads to function. So as soon as we control structure, we control function. And we very, very tightly control protein structure because that is all coded for in our DNA. Right, so these are two transport proteins, channels and carrier proteins. Here we are going to have a little bit more fun. We're going to add on a TV aerial to one of our phospholipids, because that then gives him a different name. We have here a glycoprotein. It's a bit dodgy. There we go. And this then becomes a, if you could spell it, glycolipid. So what have they got in common? They've got the TV aerial in common. And that TV aerial is standing out for a carbohydrate chain. So protein plus carbohydrate, phospholipid plus carbohydrate, glycoproteins and glycolipids. And we talked about this when we looked at protein structure, about how the protein is first made, primary, secondary, tertiary structure. Then we go on for further modifications. We can have glycolization, adding of that carbohydrate chain. So here he is in action. Okay, so these guys are used in cell signaling. They're used in cell recognition. And they're also used in membrane stabilization. Probably should have had more space for all of this. Because remember, once we have these carbohydrates, we can start getting some hydrogen bonds, and we have an aqueous environment. Okay, so you should also be getting an idea here now that we are heading towards being able to label these as the outside. You can spell outside the cell and the inside of the cell. So fluid mosaic model, we are representing our cell surface membrane. It has an outside and an inside, and those sides are different because there are different functions on the different sides. Okay, so who else have we got? We still need to talk about cholesterol. Let us have orange cholesterol. Okay, so he sits in between our phospholipids. Okay, and now we need to think back to when we were talking about saturation. Okay, so here is our cholesterol. When we talked about saturation of fatty acids, and we talked about kinks in the tail, and we said if we have a fully saturated fatty acid, then we have nice neat zigzags and they can pack closely together. If we put double bonds in, we get kinks in that tail. They don't pack so nicely together. We increase fluidity. Aha, now link that to your cell membrane. Okay, your cell membrane needs to be fluid. So let's say now we are some cell membrane in some deep sea fish in near the pole. So it's cold. Okay, so we don't want our cell membranes to freeze up. They must still be fluid. So we need to increase the fluidity of the membrane. And that is where cholesterol also plays a part. Because if you think about it, our cholesterol is there in amongst your phospholipids, right? So he's also changing the way that those phospholipids are packed. Okay, if we change the way that things are packed, we change the fluidity. What's really quite scary funky about cholesterol is that it can both increase and decrease the fluidity of the membrane, depending on how it's used. So what we say is that it regulates the fluidity of the membrane. We don't want to say that it increases fluidity, because in some cases it doesn't. We don't want to say that it decreases fluidity, because in some cases it doesn't. So keeping it to a general statement of it regulates fluidity, then we're covering all of our bases. Okay, so we have proteins, we have channels and carriers, and we have cell signaling, we have fluidity of the membrane. Okay, so again, saturation of our phospholipids would also contribute to fluidity. So let's just touch on one of these extrinsic proteins. Okay. And this is where you would want to link in with what you do for cell signaling. Okay, when we do cell signaling, it's a relatively new part of the syllabus. So you need to be a bit careful when you're learning it to make sure you get the detail. So you've got two sort of signals that can come along. You can have your hydrophobic signals, okay? Hydrophobic core, the signals can come straight through. Or you can have your hydrophilic signal, and that says on the outside, and then it causes a knock-on effect, okay? And that can link in with our extrinsic proteins quite nicely. So we can have that knock-on effect of the signal coming through, and extrinsic protein can, for example, be an enzyme. Let's actually say e.g. enzyme. So we can cause an effect on this membrane-bound enzyme from outside the cell. So when you're learning your cell signaling, you're learning a couple of pathways which are quite restricted, but it's important to remember that there's a lot more cell signaling than what you're learning. Okay, again, biology is enormous. We can't learn it all at AS, so we've got to leave some things for later on. Okay, so bear in mind you've got all sorts of things going on. We also have a cytoskeleton, which is connecting on the inside of that cell membrane, that is helping to keep the membrane and the cell shape and everything like that. But yeah, that's then all things that are going on the inside of the membrane. And that went a lot faster than I expected, right? So bear in mind that you have a 3D structure, okay? Don't restrict yourself to just 2D, but be comfortable with what is going on in the fluid mosaic model. Okay, so let's pause now and just see if we do have any questions in line with what we have already been talking about. If we don't have any questions on the fluid mosaic model, is there a question on something else? Seeing as we've still got quite a lot of time. Okay, I'm just going to check what's on the list for next week. Okay, so for cholesterol, is regulation of the membrane different to added stability? So when we regulate fluidity, we are also controlling stability. So if you're asked to give one thing for cholesterol, then you talk about regulating fluidity. If you asked to expand and describe, then I would get into stability, because the way that cholesterol is contributing to stability of the membrane is by regulating the fluidity. Any other questions? Okay, so next week I had planned to go into talking about transport across membranes, but I'm thinking that maybe we would be better off looking rather at enzymes. Because enzymes are a nice way to look at, to describe and explain, and question types. And I think that you have all covered enzymes. So let us rather have a look at that, and then we can always come back to cell membranes as we move forward. Would everybody be happy with enzymes, seeing as you guys are the regulars? You get input into this process. Or are there any other particular topics you want to add into the calendar? Okay. I think we have a lot of sleepy people this morning. Is it too early to do 9:30 on a Tuesday, ladies and gentlemen? Okay, so then it is decided we shall do enzymes. The next time we'll look at understanding enzyme reactions and linking those with the classical graphs, and then looking at that to describe and explain, because those are key question words when it comes to a lot of things, not just enzymes. So that will be next week's fun and games. In the meantime, if you have questions, then you can send them through on the chat if you are too sleepy to think of them now. Otherwise, have a wonderful rest of your Tuesday. Shall see you for sessions later, or I shall see you in the next week sometime. Have a great time, guys.